All right, here in chapter 16, looking at the second part of host defenses, chapter 15 talked about all the non-specific ways that we defend against bacteria or viruses, like our skin, complement system, those types of things. Chapter 16 is looking at how we can specifically target a particular bacteria or a particular virus. So just looking at what we refer to as adaptive immunity, meaning it can adapt, how it's different than chapter 15. So first of all, it is very specific. So again, it's going to target just one specific virus, one specific bacteria. It does allow for a lot of diversity. So one cell could react against any antigen. So again, our immune system is meant to recognize any type of foreign invader. It can also be induced, so it's not like it's on all the time. So our skin hopefully is always present, it's always there. We can't like turn it off and turn it on. However, the adaptive immune system can be kind of shut down or regulated, kind of scaled back or amplified. We can also make clones or reproduce millions and millions of cells that recognize the same invader, the same virus. So it can be amplified, and we can have tolerance to it. And so this kind of leads to the difference in whether some people react to poison ivy and others don't. So of course, poison ivy isn't actually dangerous, but in some of us, like myself, if I touch poison ivy, my body really thinks it's going to hurt me and creates a severe immune response, whereas others, they have a higher tolerance for that antigen and do not recognize it as being something harmful. But again, each cell can tolerate some level, and that varies between individual. And then the best part probably of our adaptive immunity is memory. And this is where our vaccine and kind of the theory behind vaccines, but also the idea here is that once we've been exposed to a particular virus or bacteria, some type of invader, our immune cells, once they mount a response, they store memory cells so that if we see that same invader, that same antigen, we can mount a response much faster. All right, all of this centers around our B and T cells, and these develop from lymphocytes. And here in this chapter, we'll talk a lot about antigen presentation, so what it means to present an antigen, and that gets presented on the cell surface. So we'll come back to that here in just a minute. But once an antigen, again, which is just a foreign substance, something that would trigger an immune response in a cell wall on a bacteria, or the LPS on the gram-negative cell wall, something like that that triggers, that would be an antigen. So if B cells get activated, the activation of those B cells is going to lead to the production of antibodies. So that's where our antibodies that we create come from, are from those B cells being activated. The other part of our immune system, though, uses T cells. And T cells are, instead of making antibodies, they are actually going to cause changes to our cells. And in some case, again, we're talking about a virus where that virus isn't running around in our blood, it's more inside of our cells. We would actually kill the cell in order to get rid of the virus. So it would be causing a change to the cell itself, even though it's our cell. Right, so just kind of the stages and a little difference between these B cells and T cells. So again, they both are products of different lymphocytes. So that's circulating in the bone marrow. So those lymphocytes are there. If they get activated, or as they continue to develop for B cells, those are going to go into the special sites within the bone marrow. So it's kind of easy to remember B cells, bone marrow. So B for B cell, B for bone. So that's where those are going to mature. And then ultimately, though, they enter into the lymph system. So in our lymph nodes in the spleen, which is also where those T cells are going to end up as well. However, the T cells are going to mature in the thymus. Again, kind of easy, T for T cell, T for thymus. So that's where they develop, and then ultimately, though, they migrate to the lymph nodes and the lymph system, including the spleen. So they end up in the same place, but they develop and mature in different areas. All right, so back here to what actually causes the response. So an antigen-dependent response. Again, B cells and T cells don't just randomly get activated. That would be very bad. They need to be triggered by an antigen, by a foreign substance. And again, that's called an antigen. And I mentioned that we would come back to these antigen presentation. Okay, so what happens is, because we don't want to mount an immune response, if there really isn't an antigen present, there's a checks and balance in place. So for example, on our B cells need to bind to an antigen. So there's a place here where the antigen would bind to the receptor, and it would be recognized kind of as being foreign. Okay, that's what these B receptors or B cell receptors are for, and we'll come back to those. So they would recognize the antigen. However, there's also antigen presentation, which is where a phagocytic cell, so for example, in this case, this is called a dendritic cell. Those dendritic cells would be randomly kind of taking in, phagocytosing samples of the lymph tissue. And what they do is they break down things in their lysosomes, and they place little pieces of them out on what are called MHC markers, and that's what these little things are representing. So attached to those would be a piece of this antigen. So, it gets broken down in the lysosome, and little pieces get placed on these MHC markers. And we'll talk more about those in a second too. But then what happens is if that gets activated, this is the job of the T helper cells in this case. So a T helper cell would come along, it would recognize whatever little snippet of that antigen was placed and presented on that dendritic cell. The T cell recognizes presented antigen, so not like bacteria or viruses floating around in the lymph or in the blood. It has to be presented on one of these cells. So again, like dendritic cells or phagocytic cells, that's what it's recognizing, are presented antigens. So if that T cell gets activated, depending on what type of antigen it is, so if it's something from a bacteria, then a helper T cell is going to come along, and it is going to go and it is going to confirm with that B cell that there is an invader present. So before the B cell gets activated, antigen has to bind to those receptors, and the T cell has to come along and confirm the presence of that antigen following antigen presentation. Only if both of those events happen does the B cell get activated and actually start producing antibodies. So it has to bind to the antigen, and the helper T cell has to confirm the presence of the antigen. Again, the nice thing about the adaptive immune system is that following the infection, we also have these memory B cells that are scavenging around looking for the same antigen if it comes along again, and it mounts a much faster response. Now the other part, so those were the helper T cells. If that presented antigen, though, is from a virus and it's not something free-floating, we want to activate what are called cytotoxic T cells. So that presented antigen would be placed on there, and it would bind to one of these cytotoxic T cells and activate them. And that cytotoxic T cell would then kill that cell. So again, it's toxic to cells. It would kill the cell as a way of killing the virus. And just like our B cells, we have the memory T cells, so that if that virus is presented or present in the body again, we have a much faster response. All right, and again, this is just showing kind of the rest of that slide. So those memory B cells, but also the plasma cells made from the B cells produce the antibodies, and those antibodies then go into the bloodstream to look for antigen. So again, B cells are recognizing free antigen, bacteria, or antigens hanging out in the blood or in the lymph. Whereas the cytotoxic T cells, those are going to be recognizing presented antigen. And again, our helper T cells, they help confirm the B cell and activate them. But then our cytotoxic T cells, if they get activated, they're going to kill the cell that's been infected by a virus. And there are also some regulatory T cells that just help, as the name implies, regulate and maintain the immune system. Right, so I mentioned we would come back to these MHC molecules, which just stands for major histocompatibility complex. MHC is a whole lot easier. There are two types of these, or different classes. So class 1, these are found on all nucleated human cells. So every single one of our cells has class 1 MHC molecules. What happens here are these, as our cells randomly place things from our lysosomes or degraded material on these molecules, on these class 1 molecules, so that those T cells can come along and kind of inspect. So it's a way of all of our cells kind of showing to those T cells what's going on inside. So that in the case if there is a virus inside, that's where those little virus particles would get presented on the outside of the cell so that those T cells could know they're inside. So again, all of our cells have those. We're randomly sampling the inside of our cells and placing molecules on those MHC class 1 molecules. Now, only some cells have a class 2 MHC molecule, and these are only going to be ones that can go through phagocytosis. So these are ones engulfing, again, bacteria, engulfing things from our lymph or from the bloodstream, breaking them down, and then putting little pieces, little fragments on those class 2 MHC molecules. So again, this is only in some types of white blood cells, again, those that can go through phagocytosis, and like our dendritic cells. So they're presenting, they're helping to confirm or to activate those B cells, letting them know what things are going on outside of the cell. So they're random sampling, phagocytosing things from the outside to bring them in. And then they present them so that T cells can be confirmed or activated if needed. All right, and again, this is just kind of another picture putting them both together. So B cells and T cells both have receptors for binding to antigen. It's just that on the B cell, those receptors are going to bind to antigens outside of the cell. So again, they're free floating in the blood or the lymph. Whereas with T cells, that T cell receptor is going to only recognize presented antigen. So it has to be attached to one of those MHC, either class 1 or class 2. It can't just randomly bind to things outside of the cell. It has to be presented to it. All right, just want to draw attention to this table that's in your text. It's also in the PowerPoint, but it's just a nice little summary table. I think these are nice to kind of help put things, keep things together. It's things I've already mentioned, so I won't mention them again. Just want to draw attention to it. All right, and then again, this is just kind of looking at the development of the B and T cells. So again, from the bone marrow, those lymphocytes are going to start. For the B cells, they'll mature in the bone marrow. Again, if they get activated or they go through into the lymph tissue in order to become activated, in order to look for bacteria or things floating around in the lymph tissue to become activated. Whereas again, our T cells can start in the bone marrow, but they're going to mature in the thymus, where those T cell receptors will be placed. Again, they can only recognize presented antigen. And then just like B cells, they come into those lymph nodes where they're waiting to look for the presence of antigen. All right, so just some of these interactions between our antigen presenting cells. That's what APC is talking about here, antigen presenting cell. So again, only some cells can do this. Again, dendritic cells, they're the only ones that are going to have the MHC class 2. Now they also have class 1 because all cells have those, but they're the ones. So they're randomly taking in things. So in this case, there was a microbe that's getting phagocytosed and brought in to the cell. It's going into lysosomes or it's being degraded. And as it's being chopped up and degraded, it's going to present or place pieces of that on those MHC class 2 molecules so that now T cells can come along and recognize it. And that's what we're seeing down below. So here, again, the helper T cell, once this dendritic cell is presented or placed some of that, a piece of that antigen on the MHC class 2 molecule, now our helper T cell can come along and it recognizes that presented antigen. It's not actually binding to the bacteria or recognizing it. It's recognizing a piece of it that was placed on this antigen presenting cell. That's what's going to activate it. So you can see it just a little more magnified here, how it's binding to that MHC class 2 molecule here, and then the T cell receptor are going to bind to that piece of antigen. Again, just kind of a little summary table here that looks at this. So there are multiple types of T cells, and you're not going to have to know each and every one of these. I want you to know that there are memory T cells that are stored so that a second response can be mounted faster. Those helper T cells are the ones that are going to confirm and help activate B cells so that we're getting that confirmation of an invader. And then other types of these helper cells, again, some of them are regulatory, so they kind of dampen or decrease the immune response when it's needed. Again, poison ivy doesn't really need to get activated, so those regulator T cells can kind of attenuate that response. Some of them can cause inflammation. Some can activate macrophages. But again, the ones I want you to focus on here are those helper T cells, the ones that are activating those B cells, as well as the memory cells that get activated. Right. And then just looking at how T cells become activated. Again, we have our antigen presenting cell, APC. Looking in this case, we have an MHC class 1. So this is coming from inside of the cell. So again, class 2 present antigens that came from outside of the cell. Class 2 are presenting antigens that were already inside. So again, like a virus that has infected this cell. It would be chopped up and placed on an MHC class 1. And all of our cells have these. So similar though to the class 2, the T cell receptor on this T cell would recognize the antigen presented on the MHC class 1. Now, we'll also hear the term or see CD4 and CD8. CD4 are the ones that we just went over that are going to help recognize and make helper T cells to activate B cells. CD8, those are the ones that recognize the MHC class 1, so more the viruses. So that way, that's why if you do a blood panel and they're looking for the CD4 counts or the CD8 counts, that indicates whether it's a viral infection or bacterial. If the CD8 levels increase, that indicates that it's a viral infection. It was activation by these class 1 molecules that came from inside of the cell. If it's a CD4 count that is increased, that indicates it's a bacterial infection. It was those helper T cells that are getting activated in order to activate and confirm the B cell. Right, so if there is an internal antigen that is presented and it's recognized, again, now we're going to activate that cytotoxic T cell or the CD8 cell, and it's going to create the cytotoxic T cell. And what that's going to do is it's going to release chemicals called perforins and granzymes that are going to kill the cell that it recognized. So this infected host cell is going to be destroyed by chemicals coming from the cytotoxic T cell. And again, those only get activated by those MHC class 1 molecules. Again, just a little summary table here. Again, just like with the helper, you don't have to know what helper 1 and what TH1 and TH2. I just want you to understand the general idea that the helper T cells are going to regulate or recognize the CD4, those that were presented on class 2 molecules. The cytotoxic T cells recognize those on the CD8, so the class 1 MHC molecules. All right, then just the activation of our B cells. So again, two things have to happen for the B cell to get activated. First of all, it has to bind, so its receptor has to bind to antigen. And then what needs to happen are those helper T cells need to present that class 2 molecule. So the B cell has a class 2. It's going to recognize and bind to our helper T cell, and that's going to confirm the presence of an antigen. And ultimately, the combination of those, of the B cell binding to the microbe and the cytotoxic and the helper T cell binding to the B cell, both of those events are what are going to actually activate the B cell. So we need both events to happen. Right, and again, once they're activated, we get memory B cells so that if we see it again, we know how to respond, but the majority will become plasma cells, and those plasma cells will produce antibodies. I'm not sure why exactly they draw them like snowflakes, but they're mainly showing that these antibodies kind of come together in clusters, so that as they're looking around, they can better detect the presence of antigen. So just a little bit more about the antibodies. They have 2 light chains and 2 heavy chains. They are proteins, so these are chains of amino acids. So they have this Y, ultimately this Y shape here. These chains are held together by disulfide bonds, so those are strong links. It's hard to break them apart. And then they're also divided, so the part here, kind of the ends of our Y, is called the Fab or the FAB region. This is where the antigen actually binds, so the antigen binding site. So for each antibody, there are 2 places where it can bind to antigen. And again, this is going to be different on all of our different antibodies. They're recognizing different antigens. So we have thousands of different antibodies dependent on which antigens we come in contact with. Where we can get some similarities between antibodies is what's called the FC or the F constant region, and this is what determines the class. So whether it's a class, an IgE, an IgA, all of those different categories are based, they have the same FC region, kind of the stem here of our Y. These are going to be the same based on the class. And what's similar about them is what happens once they're activated. So what effect do they have whenever they bind to antigen? And that's what this figure here is showing. So depending on the type of antigen or the that's present, or the type of, or how the antibody attaches, we have different outcomes from binding of antigen. So some of the varieties, so we can get what's called opsonization, which is what's shown here. And this is where coating of antibodies around the bacteria, around the cell, is going to activate one of those phagocytic cells here to engulf it. So this phagocyte is not recognizing the bacteria, it's recognizing the antibody attached to it. So that FC region is binding and activating phagocytosis. So that's called opsonization. It's making it more likely to be phagocytosed. So the binding of antibody causes phagocytosis of whatever is attached to the antibody. Also, in some cases, just binding neutralizes that. So if we talk about a virus, if we get a bunch of antibodies binding outside of that virus, it can't attach to a cell. So it's neutralized, it's kind of inhibited. It's not killing the virus, it's not destroying it, but it's attaching so that it cannot attach to our cells, enter in, and cause an infection. We also get agglutination, which is where antibodies start attaching kind of to multiple bacteria. So in this first one, we see that both antigen binding sites on these antibodies are attached to the same bacteria. But because there are two binding sites, what can happen is one binding site could attach to one bacteria and the other binding site could attach to the other. And if this happens multiple places, like what we see here, then what's happening is we're getting this clumping of all these bacteria together. So it's putting them in one spot, makes it easier, again, for phagocytosis. So whenever that immune cell engulfs it, they're taking in multiple bacteria. And again, it just keeps them from working. It kind of slows them down. And then we also get activation of the complement system. And then activation of the complement system depends would then determine the actual outcome. But typically what happens here with antibodies, if you remember from our complement system, when an antibody attaches and activates the complement system, that's going to cause lysing of that cell. So complement would put holes in it so that it would then lyse and rupture. And then finally, we can get an antitoxin to where the antibodies, instead of binding to the bacteria itself, it's actually binding to a toxin that was released, or a protein released from the bacterial cell. So not the cell itself, but something it's secreted like a toxin. So the antibody binds to the toxin instead of the bacteria. All right, and this is just kind of a nice little figure here that shows the importance of that secondary response, which is just the second time we're exposed to the same antigen. So you can see the first time we have this kind of long latent period because it takes a while for the B cell to get activated, get confirmation from that helper T cell, make enough plasma cells that can then produce enough antibodies to have a noticeable effect on the bacteria or on the infection. However, the second time with those memory cells, what happens is that latent period is much, much shorter because immediately once they're activated, that response occurs and we don't have that long latent period waiting for that secondary response. All right, and then again, this memory is also what sets up our vaccines. So the idea behind a vaccine is that you are exposing somebody to either the whole virus or bacteria or a part of it, so that we're activating the immune system to create those memory cells. Now, how much of an immune response we get determines how long the vaccine lasts. So if we mount a huge immune response, then those memory cells, we don't have to typically have a booster, or the time in between boosters is really long. Whereas with other vaccines, we have to get kind of annual or more frequent boosters because the immune response was activated, but it's not long lasting. It wasn't a robust response that led to a lot of memory cells being created. So what this kind of summary figure here is showing, so we have whole organism vaccines. So this is where the entire bacteria or the entire virus is added to the recipient, but in all cases, something has been happened. We're not causing an infection. Something has happened to that whole cell. So in this case, in 1A, the cell has been killed or the virus has been inactivated. So inactivating a virus would be giving you the whole outside of the virus, the capsid and the protein coat, but removing its DNA or removing something needed. So even though it's in the body, it can't replicate. or again, killing a cell. So this bacterial cell here, the whole thing is still being added, but it has been heated or killed so that it cannot replicate. Live attenuated, so these are still, as it says, alive. They could reproduce. However, they've been hindered or weakened in some way to where they reproduce much, much slower, and it gives our immune system plenty of time to mount a response so that we get a great immune response, but without getting that infection. So these are the ones that are going to cause that robust immune response, and the boosters are much less frequent. And then the last one here, so adding kind of a third party, kind of harmless virus that has DNA or RNA from a surface protein. So we're giving somebody a virus containing a harmless virus that contains information that triggers an immune response. And this is what happens with the COVID vaccine. So it's mRNA that's packaged into a harmless virus, but whenever that virus infects the cell, our cells, the cell produces a capsid, a protein from the virus. It doesn't make the whole virus, so it can't cause a COVID response, but the presence of that capsid, the presence of that viral protein, mounts an immune response. And then finally, just some of the kind of antigenic components. So these are not whole cells or whole viruses. So again, just giving the surface antigen, so just giving somebody the capsid would be different than packaging the DNA or RNA needed to make the capsid into a virus and injecting it. So here with these antigenic components, it would just be adding part of the virus or part of the bacteria that would cause an immune response. Also, mRNA can be packaged inside of a lipid vesicle. So instead of inside of a virus, it's just put into a lipid. Same idea though. That lipid then goes into the cell. The cell creates a protein based on that genetic information. All right, and then our other one here, the antigen complexed to a protein. So taking that same surface antigen like a capsid, but attaching it to a protein, that's going to help trigger an immune response. Again, the protein isn't harmful, but it helps to cause and activate the immune response. And these are called conjugated vaccines. All right, that one's a little longer this time. Lots to unpackage there with our adaptive immune system.